So I'll be your host today. My name is Alex Podcat. I'm an invasive species project coordinator. Today we're going to hear from Saanchi Singh. She's a graduate student with the Canadian Food Inspection Agency and a student at Carleton University. And she'll be speaking about using genomic tools for invasive seed detection. The Invasive Species Center respectfully acknowledges that our head office is located on the traditional lands of the Anishinaabe, the Batchewana and Garden River First Nation, as well as the longtime settlement of the Métis people in the Robinson Huron Treaty area. The Invasive Species Center is a not-for-profit organization that mobilizes action against invasive species that harm the environment, economy, and society. We're based in Sault Ste. Marie, but we have staff kind of across Ontario. I wanted to switch to 5. There you go. We also have free resources on our website. We have a lot of species profiles and best management practices, as well as much more. And we have our newsletter. So you can sign up for our newsletter and stay up to date on events and more on our homepage of our website, invasivespeciescenter.ca. We also have our training program, the Invasive Species Center, the Invasive Species Training Program, which is a first virtual self-paced program that offers courses on invasive species. I believe we currently have 5 training programs with more on the way. So it's my pleasure to introduce Saanchi. She is a graduate student at the Canadian Food Inspection Agency and Carleton University. Saanchi's research focuses on invasive plant seeds and their ability to contaminate crop seeds, reducing harvest quality and causing significant economic loss. Early and accurate identification of these seeds is crucial to prevent their spread. At the Canadian Food Inspection Agency, they are developing 2 DNA-based methods for accurate and efficient invasive plant detection: DNA metabarcoding and DNA macro-barcoding. So we'll be hearing more about that from Saanchi. So thank you for being here, Saanchi. We'll get you to start sharing your screen. While you're getting that going, I'll just go over some housekeeping with everyone. If you could put any questions that you have about the presentation in the Q&A section, the Q&A box, rather than the chat, just to keep things organized. And then I'll read the questions at the end, and we'll go over them then. We also have a feedback survey at the end for you all to fill out for us to review. And with that, I'll pass it over to Saanchi.
Can you see my screen?
Yes.
So thank you so much. I'd like to first extend a big thank you to Alex, Taylor, everyone else at the Invasive Species Center for coordinating this and allowing me to nerd out a little bit about invasive seeds and plants today. So my name is Saanchi. I'm a graduate student at the Canadian Food Inspection Agency, CFI in short, specifically part of the Molecular Identification Research Laboratory here, and I'm also part of Carleton University here in Ottawa. And today I'll be talking about genomic tools, or some of them that we're developing for invasive seed detection so that we're no longer lost in the weeds. This is a little bit of an overview of what I'll be talking about today. So first, I'll give you a little bit of context in regards to Canadian trade and agriculture, followed by the CFIA's role in invasive and noxious seed regulation, followed by some of the species that our lab is focusing on and working on currently. And then I'd like to give a little bit of context with regards to some of the conventional methods that are used currently with regards to seed regulation. And then I'll delve right into what we're developing, the DNA-based tools, so the metabarcoding and macro-barcoding. I'll also take some time to talk about some future directions of this project. And then finally, how you can use these tools for your projects and your research. So trade and agriculture is extremely important for the Canadian economy, specifically the production of wheat and canola. Canada is the largest producer and exporter of canola in the world. 90% of all canola is produced in Canada, is exported, and these exports are valued at around $16 billion. Wheat is the largest field crop that is grown in Canada. Wheat exports are valued at around $9 billion, making Canada the third largest exporter of wheat in the world. But one of the major threats to this billion-dollar industry are invasive plants and weeds, whose seeds can then get mixed into grain shipments or harvests, or whose seeds can, hitchhike on vehicles, equipment, clothing, livestock, or more. And ultimately, all of this costs $2 billion annually in management, and this cost is expected to rise with the proliferation of invasive species and plants as the climate warms due to climate change. So the CFIA is Canada's national plant protection agency that ensures that all seeds sold and exported in Canada comply with international standards. The CFIA is responsible for regulating the spread of noxious weeds, and these are weeds that are regulated through the Seed Act. And for a plant to be considered for regulation, risk assessments are performed to identify a plant's ability to spread and establish populations and its risk to Canadian agriculture or ecosystems or human activities. So I thought I can give you a little bit of context regards to how weeds are classified as noxious. I'll be focusing on the 3 classes, even though there are 6 of them, because the species I'll be talking about later on fall into one of these 3 classes. So we have class 1. These are prohibited noxious weed seeds. This means that seed lots must be free of these seeds. These weeds can either not be in Canada or they are under the official goal of eradication. We have class 2 weeds, which are primary noxious weed seeds. And these are present in Canada, but they're not widespread yet. And so they're not an official target eradication yet. And then we have class 3, these are secondary noxious weed seeds. They are present in Canada, but they are permitted in restricted quantities. So class 1 are the worst ones by regulatory standards, and as the class number increases, you can think of the weed potency decreasing. So there are many noxious species that impact the quality of Canadian canola. I'll be talking about a few of these noxious species, specifically the ones our lab is working on, and what characteristics make them problematic weeds. So up first, we have Agelop cylindrica, commonly known as jointed goatgrass. It is a class 1 noxious weed that grows with wheat, and it shares one of its genomes with wheat. And genes for herbicide resistance are found on that shared genome, which means that there's no selective herbicide for jointed goatgrass. And so if you're spraying herbicides to control jointed goatgrass, you're effectively killing the wheat along with it. And then we have Avena fatua, commonly known as wild oat. It's the class 3 weed, and it impacts canola and wheat, both of them. It has a longer period of emergence compared to these crops, meaning that it can emerge earlier or later, and both are advantageous for wild oat. So if it emerges earlier, the seeds shed earlier and they can persist in the soil. If it emerges after either of these crops emerge, then wild oat can bypass herbicide application and continue to grow, produce seed, contaminate harvests, or contribute to the soil seed bank. And on top of that, wild oat is becoming increasingly resistant to herbicides. We're also working on Gallium spurium, which is commonly known as false cleavers. This is a class 3 noxious weed. It impacts also canola and wheat, both of them. It's unable to stand on its own, so it uses these little hairs, which I've circled in the leftmost image, called trichome hairs. And it uses these trichome hairs to stick to other plants, livestock, and equipment. It also has these extremely long trailing stems that can get stuck in farm equipment. So you can think of its mechanism of action as being mechanical. And we're also working on Sinapis arvensis, which is commonly known as mustard. It's a class 3 weed. It's also a prolific seed producer. Each plant can produce at least 3,000 seeds. It also grows taller compared to nearby plants, and its leaf area is larger than nearby plants. And so this provides neighboring plants from photosynthesizing. And in fact, studies have shown that it can reduce canola yields by 36% when densities of wild mustard are as low as 20 plants per meter squared. It also has large seed banks, which can remain viable for 60 years. So again, very problematic. And we're also working on weedy pigweed species, and we're specifically focusing on Amaranthus tuberculatus, commonly known as the tall water hemp, as well as Amaranthus palmeri, which is commonly known as Palmer amaranth. So both of these species are prolific seed producers again. Each plant can produce up to 1 million seeds, and they're both resistant to a wide variety of herbicides. The tall water hemp is regulated in Canada and is a class 2 noxious weed, and it has populations that are established in Ontario, Quebec, and Manitoba. Palmer amaranth, on the other hand, is not regulated in Canada, and it's not regulated federally in states as well, but there are some states which regulate it. Down south, some like Iowa, Ohio, Minnesota, and a couple others. And even though Palmeri is not regulated in Canada, it has the potential to expand northward, as seen in the map on the complete right, showing the potential range expansion of it. And in fact, Palmeri populations have been detected in Manitoba and Ontario already, so it has the potential to spread further. And so that leaves us with the question, what can we do? Well, the most effective strategy is to prevent their introduction. So from this graph, we see that as time goes on, time seen on the x-axis, the area infested with weeds increases and the cost to control them also increases. And as time progresses, different strategies need to be implemented to control growing populations of weeds. But again, the most cost-effective strategy is to prevent their introduction into our agricultural systems to begin with. And since seeds are the primary way that invasives and noxious weeds travel long distances, especially through trade routes, preventing the introduction of seeds is especially important. And so, how do we prevent the introduction of noxious seeds? Well, we have to be able to identify them. So conventional methods of identification are based on physical characterization. So we want to be able to distinguish between noxious seeds, such as wild mustard, shown here on the left, and commodities like rapeseed, which is similar to canola, shown here on the right. And so I'm sure we can all appreciate just by looking at these two images, it's incredibly hard to spot the difference even when we're zoomed in so much. We also want to be able to distinguish between closely related species, which may not be noxious, and in fact, they may have some economic value, as in the case here with common oats, which is what we eat in oatmeal for breakfast every morning, as seen here on the left, compared to Avena fatua wild oat on the right. And here I'm showing you images that were taken from our microscope in our lab. I'm going to go grass on the left, wheat seeds on the right. And although they look very dissimilar when zoomed in this much, I'm sure you can appreciate the difficulty in identifying jointed goat grass from thousands of wheat seeds, or as is the case in this image, among 499 wheat seeds. And this image was again taken in our lab. So you can imagine that, you're a certified seed analyst, you're sitting at your microscope, you're sifting through 25,000 background seeds to identify that one jointed goat grass seed, because that's what you're required to do by regulatory standards, to identify that 1 in 25,000. And not only are there commodity seeds like wheat in there, but there may be close relatives of that noxious seed in the mix. And so now you have to distinguish between all of them, and you're doing this based on just a few physical characteristics. So I think we can all safely say that physical characterization of seeds is technically challenging, even if you're an expert, and this can be time consuming. And so our lab is developing 2 DNA-based methodologies to complement these conventional methods. The first one that we'll be talking about today is DNA metabarcoding. And I like to just take a few steps back and talk about what DNA barcodes are. So DNA barcodes are similar to grocery store barcodes in the sense that they both enable us to uniquely identify products like orange juice, or in the case of DNA barcodes, species, simply based on a few characteristic differences in their barcodes. So there are certain gene regions that contain characteristic differences in the DNA sequence, which corresponds to specific species. So here in the figure that is labeled part of the DNA barcodes, there are unique differences between grass species that I'm showing you with the arrows. And if it helps, you can also think of DNA barcodes sort of like fingerprints. So what happens with DNA barcoding is that you take your sample, you extract DNA from it, and then you amplify gene regions of interest that you know contain enough differences between species. So in the plant world, these are usually RBCL, MAC-K, ITS2. And then these DNA fragments are then sequenced, which basically means that these fragments are read serially to determine the sequence of nucleotides, so the patterns of A, T, C, and Gs in that barcode. And with DNA barcoding, you can identify one species at a time. With DNA metabarcoding, it's a similar process. You start off with DNA extraction, you amplify the gene region of interest. But you can identify multiple species at the same time using high-throughput sequencing methods, which allow you to massively sequence in parallel on a large scale. So each step in this process, from the DNA extraction, the PCR, the sequencing, it can be standardized, which would make this methodology a good addition to the current regulatory framework. So I'll get my laser pointer out for this one. I think that'll be helpful. But we barcoded 10 species of Agelops. One of them was Agelops cylindrica, so jointed goatgrass, and the other 9 being close relatives of it, along with wheat. And this is just to ensure that we can uniquely identify jointed goatgrass from everything else. So we barcoded the ITS2 and RBCL regions. So in the ITS2 region, we saw that jointed goatgrass, which is up here in this blue box, had the exact same variants as one of its close relatives, Agelops transialis, which is down here in this black box. And so we weren't able to uniquely distinguish jointed goatgrass. And so we had to use RBCL to be able to distinguish jointed goatgrass from Agelops transialis. So if we see down here in this black box, transialis has these T's here, which are green, and jointed goatgrass doesn't have those T's there. And so with a combination of ITS2 and RBCL, we are able to distinguish jointed goatgrass from close relatives. But for the rest of the presentation, we're going to be focusing on jointed goatgrass and wheat. And for this, we only need ITS2 to reliably distinguish between the two, as seen by the species-specific differences here. So I know this figure is a little complicated, but I'll break it down a little bit. We performed a limit of detection experiment to determine what is the limit of detection of this technology. So for that, we spiked wheat seeds with jointed goatgrass and increasing dilutions. We had 3 different test conditions. We had a test condition, a positive control, and a negative control. In the test condition, we mixed 1 jointed goatgrass seed in 99 wheat seeds for a dilution of 1 in 100. We also mixed 1 jointed goatgrass seed in 249 wheat seeds for a dilution of 1 in 250. And we mixed that 1 jointed goatgrass seed in 499 wheat seeds for a dilution of 1 in 500. These mixes were ground up and then DNA was extracted. For our positive control, we ground up the jointed goatgrass seed and the wheat seeds separately from each other. And then the DNA extraction we performed separately, but then we mixed the DNA together to reflect the same dilutions that I just mentioned. And then for each dilution, for the test condition and the positive control, there were 3 different accessions of jointed goatgrass, and they were used in triplicate. Finally, for our negative control, we only had wheat seeds, and again, these were present in the same dilutions as earlier, and DNA was extracted. So following this, the ITS2 region, which I just showed, was amplified. And then following that, an index PCR was performed where unique identifying DNA tags were added to the ends of the ITS2 regions for each sample. And this allows everything to be pulled together and then sequenced at the same time. So this is where the high throughput part of the technology comes in, because you're still able to determine which sample is which, since each DNA tag corresponds to a different sample. And these were the results of that run. These are normalized reads. These are the average of the 3 replicates for the seed mixes and the positive DNA control mixes for the reads that belong to jointed go-grass. So the lightest blue color here corresponds to the lowest dilution of 1 in 100. That medium in between shade of blue corresponds to a dilution of 1 in 250, and then the darkest blue corresponds to the most diluted jointed go-grass sample of 1 in 500. So there's a few things I want to highlight from here. First off is that we saw that as the target seed got more and more diluted, we see fewer and fewer reads corresponding to jointed go-grass. And this is expected since that seed is getting further and further diluted in wheat seeds. But despite that, we were able to identify that one jointed go-grass seed in a dilution of 1 in 500, which we were very pleased and happy with for our first run. We also saw that the seed mixes, which are highlighted in the dotted lines, they had fewer reads in comparison to their DNA positive control counterparts. And this is also expected since everything's getting ground up together and you can't really control how well that one jointed go-grass seed is getting ground up and therefore how much DNA is extracted from it. We also see that the seed mixes had more variation because of the larger error bars compared to their DNA counterparts. And this also, again, makes sense because it is a less controlled environment, so you are expecting more variation in the amount of DNA that you're getting and therefore the amount of reads. And finally, we saw that the negative control had no reads corresponding to jointed go-grass, which also makes sense because the negative only contained wheat seeds. And so currently we are continuing to increase the dilutions to determine the true limit of detection of this technology and to see if we can get to that 1 in 25,000 that is required by regulatory standards. We are also performing the same spike experiments with other Noxie species that are regulatory concern that I mentioned in the very beginning. So these include wild oat, wild mustard, and pulse cleavers. And as I mentioned in the beginning, we are testing 2 different DNA methods. The other method that we're testing or calling and piloting is macro-barcoding. And for this, we're using amaranthus. So there are multiple types of DNA barcodes. There are conventionally sized barcodes, which we've been talking about so far. So things like RBCL and ITS2. These are smaller. They are around 300 to 700 base pairs long. And then we have super barcodes, which cover the entire chloroplast genome, so in magnitudes of hundreds of thousands of base pairs. Macro barcodes, or what we're calling macro barcodes, contain multiple gene regions that can be sequenced in one long fragment using long-range sequencing technologies like nanopore, which allow you to sequence longer and longer fragments. This circumvents the need to optimize multiple PCR reactions from multiple gene regions, which is often required for plants because there is no universal plant barcode. And you also don't need large amounts of high-quality DNA, which is often required for super barcodes. So I like to think of the macro barcode sort of as the Goldilocks of all the barcodes. So this is a process that we took to developing the macro barcodes. So we first identified a 5 kb region in the chloroplast that contains enough differences between species to act as a barcode. We then extracted DNA from our amaranthus species, and we amplified that 5 kb region via PCR. We purified that PCR reaction, and then we sequenced our samples and our 5 kb fragments using nanopore sequencer called the MinION, which fits in the palm of your hand. So that's really cool. And following this, we performed a quality check on the data. And in this case, there were thousands of reads per sample. And so we filter them based on the expected length and also other quality control metrics. And then following this, we mapped our reads, which meant that we aligned them to the exact same 5 kb region in the chloroplast, depending on the species. And since we had thousands of reads per sample, we generated consensus sequences, which essentially means that we took the most average or most common nucleotides at each position of the barcode. And then these consensus sequences were then aligned to identify species-specific differences. And so I'm showing you a little bit of the alignments here. So out of the 24 samples that we macro-barcoded, which corresponded to 13 different species of Amaranthus, we were able to uniquely identify 11 of the 13. We were unable to distinguish Amaranthus palmeri from Amaranthus watsoni. And this is not surprising, they're sister species, and they only have 2 differences across the entire class. But here I'm showing you the 11 of the 13 that we were able to distinguish, and they're identifying regions, and the exact location of those regions and the alignments are shown up below them. And so eventually, we're going to be expanding this macro-barcoding into other species like jointed goatgrass and fold this macro-barcoding into our metabarcoding pipeline. I also wanted to give a quick little summary of how each type of barcode performed. So with the super barcode, we were able to identify all 13 species, one barcode, one super barcode. With the macro barcode, we were able to identify 11 species with that one macro barcode. And we also tested conventionally sized barcodes, and we were able to distinguish all 13 species with 2 barcodes. However, we first had to test 7 different conventionally sized barcodes, optimize conditions for each of them to only end up having to use 2 for species distinction. And so ultimately, less optimization for the macro barcodes was required and less input DNA was needed in comparison to these super barcodes simply due to that medium size. And so now I'd like to take some time to talk about future directions of this project, like what we're working on currently or planning on working on soon. So we're working on detecting noxious weed seeds in the soil. And so weed seeds can persist in the soil seed bank for years upon years. The soil seed bank is dynamic, so some seeds will decay in the soil, some will persist but not be viable, some will germinate and emerge, some will get eaten by birds, insects. So there's a lot of things that are happening in the soil in relation to seeds. You can also think of seed dormancy itself as being dynamic. So seeds can go from being dormant to non-dormant depending on environmental conditions. And seed dormancy is in fact one of the biggest barriers to weed control. In fact, less than 10% of wheat seeds germinate each year. So you can imagine after each growth season, how many seeds are accumulating in the soil. And so preventing weed seeds from setting seed is the most effective management strategy in this case. And this is commonly done using herbicides, but the most effective herbicide application is species specific. And for that, we need to be able to determine the identity of the weeds in seed form. And so in order to be able to determine the identity of weed seeds, conventional methods rely on, again, physical characteristics, but as we know, this is challenging, especially if they're extremely small, if they're closely related. And if seeds are indistinguishable, then they're grown, but this can take weeks or months depending on the species. And this further prolongs a time-sensitive process and it prevents in-season agricultural management decisions from taking place. So we're working on a DNA metabarcoding approach as a way to complement these conventional methods. The first process would be to filter out the seeds via sieving, and then also using flotation to separate out the seeds from soil simply based on density. And then once we have our seeds isolated from everything else, we'll go through the process of doing metabarcoding. So things like DNA extraction, PCR, and then sequencing. So we are currently in the process of developing a protocol to filter out the seeds. So we're in that initial stage of this process. And we're doing this in collaboration with researchers from the Institut de Recherche de Développement en Agro-Environnement in Quebec. And we anticipate this DNA metabarcoding complementary approach to take up to a week at max. We're also working on developing molecular seed viability tests because even if there are noxious seeds that are present in a seed lot. If they're not viable, then it doesn't really matter because they're unable to grow and therefore propagate. So we need to be able to identify the identity of the weed along with its viability. So as I mentioned earlier, weed seeds can remain dormant, and staggering germination times keeps the plant safe from possible weather events, herbivores, or in the case of weeds, herbicide application. So it is a survival mechanism. Seeds will naturally break dormancy by a process called invasive. And so seeds will first take up water, which will soften and swell that hard seed coat. And then this uptake of water will activate any internal physiology. So the cells will start to respire, the seeds will start making protein and they'll metabolize their food sources. And a primary root will break through and start absorbing water from the soil. And then the cells of the seed will continue to divide, elongate, and grow, leading to the formation of a seedling. And so conventional methods for seed viability, there are two of them that are currently used or frequently used. Germination tests are the most accurate way of determining seed viability. This can take weeks, and if dormancy is suspected, you have to break dormancy effectively. And this is again because just because a weed is unable to grow, it doesn't mean that it's not viable. It could just be simply dormant. And so dormancy is usually broken through techniques like pre-chilling or mechanical scarification in which the seed coat is chipped. And with tetrazolium tests, you're measuring the activity of dehydrogenase enzymes that are present in metabolically active cells. So if the tetrazolium dye interacts with the hydrogen ion, which are released by these enzymes, then the seed will be stained red, indicating that it's viable. And if it's not stained red, then that indicates that it's not viable. But seeds can be partially stained, and so this partial staining can be misinterpreted. This can lead to false negatives. And so we're using more of a molecular approach with regards to seed viability. So unlike for species identification, DNA cannot be used for viability testing since DNA can remain intact even in non-viable seeds as it's pretty stable and it degrades slowly. It's a pretty stable molecule. However, RNA is present in metabolically active cells. And if the cell dies, then the RNA degrades very quickly, meaning that it can be used as a viability indicator. And so with the current state of the project, we need DNA barcodes to be able to identify noxious species and RNA markers for viability. So eventually we want to be able to find RNA markers that allow us to do both concurrently, determine the identity of the seed and also use for viability. But that's like finding a needle in a haystack. And so we're taking things one step at a time. And so our first step has been to find RNA markers for viability. So all of this preliminary testing that I'm showing you here today was done by Steffi, who was a student at CFIA in our lab. And so from literature, we identified that abscisic acid could be a potential viability marker. And this is because it's constantly expressed through different seed stages, so from dormancy to germination. And abscisic acid is a plant hormone that regulates seed maturity and germination. So this means that if a seed is alive, even if it's dormant, it should still be expressing these abscisic acid genes. And we should be able to detect viability even if it's dormant. If the seed is dead, then the RNA from these genes will quickly degrade and that expression of abscisic acid will be absent. And so Steffi, what she did was she was able to identify the presence of one of these abscisic acid genes called ABI3 with two sets of primers, and that's what I'm showing you here in the left image. For Amaranthus palmeri and tuberculatus, you can see the presence of this gene with the bright white bands on that gel. And so currently we're performing some data analysis. We ran an RNA-seq experiment, and that allowed us to obtain RNA profiles for dormant seeds and seeds undergoing inhibition. And this will allow us to find all the possible candidate viability markers, and so that we're not just listed or restricted to the RNA markers that were listed in literature. There could be stronger markers, and this will allow us to identify those. And that is currently happening. So there are three key takeaways I like to leave you with today. The first being that DNA metabarcoding is sensitive enough to detect noxious weed seeds from bulk seeds using high throughput sequencing technologies. DNA macro-barcoding is a good, feasible compromise between size and resolution, especially with the increasing use of long-range sequencing technologies like nanopore, and that you are able to use these DNA approaches too for your projects. So I went through the Invasive Species Center's website and I looked at some of the most common invasives that are being worked on in Canada. And I've pulled some of these. And a potential application for the technologies that I showed you today could be to distinguish between invasive Phragmites from native Phragmites, which are visually hard to distinguish, especially in the summer, as seen from the leftmost image and the rightmost image. You could also use these approaches to distinguish kudzu, the invasive kudzu, the vine that is the south from species like the hog peanut or the trailing wild bean in their early life stages, which is when it's extremely hard to distinguish between these species. Or you could use these to distinguish seeds. So kudzu seeds from Saanchi seeds, which to me right now look pretty similar looking at the rightmost image. And you could also use these approaches to distinguish between giant hogweed and cow parsnip, where one of the main visual differences is the size. And as the name suggests, giant hogweed is extremely tall, especially in comparison to cow parsnip. And by the time these species mature and reach that distinguishing height, populations have started to establish. So DNA approaches can be used. Or could be applied during the earlier life stages where it's harder to distinguish the species. And with that, I'd love to acknowledge everyone who has worked on these projects and for their guidance and their collaboration. So everyone at Merle, at the CFIA, my supervisors, my committee members, collaborators, and of course funding sources. And after this webinar, if any of you have any questions, please email me, and my email can be found on the screen. And I'll take any questions that you may have.
All right, thank you so much, Saanchi. That was a great presentation. It's very, very in-depth and informative, very interesting stuff. So I'll read out any of the questions you guys have. If you want to throw any more questions in the Q&A box. First up is zooming out a little bit. Someone's wondering, how are invasive species introduced to a new environment?
It can be many ways. If we're thinking about more of like a Canada-wide or global perspective, it's usually through trade. So you'll have like invasive seeds that can get mixed in with crop trades. And so that's an easy way for weed seeds or invasives to get mixed in and then get and propagate within Canada. In terms of like within Canada, like between provinces or even between certain regions, seeds can, hitchhike on clothing, equipment, or whatever it may be. And then wherever they are, they can establish and they can propagate that way. So it seems like seeds are a very big way that invasives travel and propagate and get introduced to in Canada.
All right. Are there any specific crops that are not currently considered that might be a good choice for this program?
I think because of the CFIA, we are focusing specifically on regulated species because of the mandate. But I'm sure you could apply these to any of the other species, like the ones that I mentioned. It's a very versatile approach because it is DNA at the end of the day. But at the CFIA, we are required to focus more on regulated species.
Okay. Are there any species outside of agriculture and plants that are considered for this program?
So again, with the CFIA, we have to look at what's regulated and what's not regulated, and most of the weed species or invasive species are part of impact agriculture, like the ones that I mentioned, but there are also some, I'm sure, that impact forestry, because that is also a big industry in Canada. But we focus specifically on agriculture or more on agriculture. And we also have things like Cascuda, which impacts wetlands as well. But again, we're focusing more on regulated species.
Right. If I encounter an invasive species in the wild, what should I do?
The first thing would be to contact the CFIA and let officers know that you found the species there, and take pictures of it. Make sure that you're not, having its seed spread anywhere. But the first step would be to contact the CFIA about it.
Okay. Are there any regulations for diagnosis labs wanting to use these tools to offer identification services?
So the tools that we're working on right now, they're not, we haven't gone to that stage where diagnostics is able to use them. That's what we're working on towards, where we have to do a lot more testing. So specifically with the metabarcoding, determining the limit of detection to see if we can get to that 1 in 25,000. And so once we get to that stage, then diagnostics can use these tools to complement the conventional methods that are used. But for that, we need to go through the process of standardizing these technologies and making them pretty solid.
Okay. Does the CFA have a service to process samples or to submit priority species for development of these processes?
Um, do you repeat the question, sorry?
Does the CFIA have a service to process samples or to submit priority species for development of these processes?
Yeah, so we have a seed lab in Saskatoon that actually goes through these species, or goes through seed lots to be able to identify them. And we also have a Genobot lab here in Ottawa that also does some testing, so you can submit samples to the CFI in that way. Um, Again, the methods that I'm showing you today, they won't be used for that process because we have to still standardize these processes and these methodologies. But they use current regulatory frameworks methods or methods that are um, standardized globally to identify species, and you can send it to them.
Great. Are there any opportunities for labs to collaborate on this project?
Oh, absolutely. We're always looking for ways to collaborate, whether that be to expand the species that we're including in our project or in these technologies or to develop new technologies. You're more than welcome to email me. My email is found on that screen and we can continue the discussion that way.
Okay, and has citizen science applications such as iNaturalist helped in this research?
A little bit, yeah, for sure. So when we get samples, sometimes they're not the samples that we get, sometimes it could be plant matter. Sometimes it's difficult for us to identify them, or they say that they're certain species, but they're really not when you look at their DNA. And so from that, we go through iNaturalist and we can see what people have reported certain species as, and we can compare them that way visually. And so, yeah, iNaturalist definitely has a big role that plays in what we're doing.
Are there any new species that are recently on your radar with regards to this program?
New species, I think that would be Amaranthus palmeri because it's not currently regulated. And so that's really on our radar because we can see that it's expanding. It's already in Ontario, Manitoba, and Quebec, and it's just time before it spreads more. And so that's the species that we're also focusing on despite it not being regulated.
Okay. Okay, that seems to be um, all the questions at the moment. We'll wrap up. Um, Thank you again, Saanchi, for presenting and answering all of the questions that were asked. We really appreciate you joining us today. And thanks to everyone for listening. You can sign up to our newsletter to stay up to date on future webinars and events. And if you have any more questions for Saanchi, you can email her. So thank you all again for joining us. Thank you, Saanchi, and have a great rest of your day.